discussing our newest sequencing panel, our TruSeq Bovine Parentage panel and Sequence Genotyper Analysis tool. By the end of this webinar, you should know how the TruSeq Bovine Parentage panel generates sequencing libraries, recognize the steps that are important within the workflow and are critical for successful library prep, and understand how to use the analysis tool Sequence Genotyper for downstream analysis. So just want to give you a brief introduction as far as to the TruSeq Bovine Parentage panel. I'm going to cover a couple of topics. One of them is, what is parentage and why is this important? Also, I will be discussing what the specifications are for our TruSeq Bovine Parentage panel. So what is parentage and why is it important? Parentage is examining the DNA from an offspring and a parent, and they're compared to estimate the likelihood of the two individuals that are being related. Basically wanted to know who are the parental units of the specific offspring. Knowing parentage is very important in agricultural research because this accelerates genetic improvements by increasing pedigree accuracy. One major contributor to agricultural research is the International Society for Animal Genomics, and this group focuses on basic and applied research within multiple fields of genetics, and they're influential in the community and the content developer for a variety of parentage testing. So knowing parentage and knowing pedigree is very important, like I mentioned, in agricultural research because it can help you discover high-value calves within a herd. It can also provide great tools for information for breed decisions, and also, again, linking back to pedigree accuracy and accelerating genetic gain. So some of the specifications for our TruSeq Bovine Parentage panel are listed here. The content is comprised of 263 SNPs. 200 of them are variant targets associated with with parentage assessment. These are all ISAG, or International Society of Agricultural Genomics, targets, SNPs that have been identified. The rest of the SNPs within the panel that are covered are variant targets associated with genetic disorders and diseases, like congenital muscular dystonia, cardiomyopathy, hemophilia, and also variant targets associated with economical value traits like male fertility, meat, and milk quality. The assay, you have enough reagents to generate 96 samples or 96 libraries using our TruSeq Custom Amplicon version 1.5 library prep workflow. The kit includes 96 unique indices, which we'll be discussing later on in the talk. You have the capability of pooling all 96 samples in one MySeq run using V3 chemistry, and the run is configured as a 1x121 run. Once the sequencing is completed, we then use our Sequence Genotyper tool to convert the sequencing data into genotyping calls. It's a web-based desktop application, and we'll be discussing that later in the talk. As far as specifications, we would expect to see uniformity at about 97%, coverage of the ISAG targets at least 100x. We should be seeing anywhere between 98 and 100%. We should be seeing 100% coverage of all of the ISAG targets, very reproducible between 99 and 100%, and also very accurate genotyping calls between 97 and 99%. And so when you order the TruSeq Bovine Parentage panel, what do you get? So you get a few components here. One of them will be the reagents necessary to generate the library. This includes the oligo pool, that are the probes that are directed towards these SNPs, and the rest of the reagents to generate the libraries. You also will be receiving the MySeq V3 sequencing cartridge and reagents, and flow cell included, 150 cycle kit. And you also will be able to go to our support site and download the Sequence Genotyper Analysis tool, as well as MySeq Reporter that are used in conjunction for downstream analysis. So I mentioned earlier that the TruSeq Bovine Parentage panel utilizes our very established TruSeq custom AFRICOM workflow. And here on the right, I have a brief overview of what the steps to generate a library using this workflow consist of. First, we hybridize oligo probes to the areas flanking the regions of interest, in this case, the SNPs that we're interested in targeting. We then proceed with an extension ligation that creates an AFRICOM template. From this AFRICOM template, we then proceed to a PCR step that adds on our dual index barcodes as well as finishing off the adapter, at which the end you'll have a targeted AFRICOM library that is ready for cluster generation and sequencing. So this is, like I mentioned earlier, a brief overview of the workflow. If we take a deeper dive into the workflow, what do each of these steps consist of? So first, we start off with quantifying the genomic DNA. We proceed to hybridizing the oligo pool. We then remove all unbound oligos, and then we proceed to the extension ligation step, followed by PCR amplification that introduces the dual index barcodes. We clean up the libraries using AmPure beads. We then normalize the libraries, pool the libraries, and then proceed on to the MiSeq for sequencing, and then once sequence is completed, on to data analysis. So if you go to the first step to quantify genomic DNA, we want to make sure that we're using a fluorescent-based method to quantify the DNA. In this case, we either recommend pico green or qubit reagents. For this particular kit, we recommend that 50 nanograms of high-quality genomic DNA is used. You can always check the quality using a nanodrop measuring the 260-280 ratio, and it should be anywhere between 1.8 and 2. Once we have our genomic DNA diluted to the appropriate concentration, we then proceed to hybridize the oligo pool. In this step, we incubate the genomic DNA with the oligo pool. We first, on a heat block, we preheat the heat block to 95 degrees, and then we set it down to 40 degrees, and this allows for a gradual cool down. And this gradual cool down is key to successful hybridization of the oligo pool, and now you will have the probes binding to the specific areas within the genome. This typically takes around 80 minutes, give or take, but the temperature is more important than the time. At this point, once we have the samples hybridized to the oligo pool, we then move the samples over to a filter plate, which will then remove the unbound oligos. This is a little bit different from a lot of our different library preparation kits. We are using a filter plate. And so what does this filter plate unit is comprised of? Here I have a schematic of it on the right-hand side. You have a lid, you have the filter plate, a metal collar that some customers find useful to help align the filter plate with the MIDI plate collection plate at the bottom. One key thing is that we want to pre-wash the filter plate. And this is very important because it increases the DNA hybridization, and it also kind of helps you assure that you're getting proper filtration through all of the wells within the filter plate, and identify any wells that are not filtering as expected. Once the filter plate is pre-washed, we then add our sample to the filter, centrifuge, and this will wash to remove the unbound oligos. While the sample is still on the filter, we then proceed to the extension ligation of the target region. And in this case, we have the extension ligation reaction creates an AFRICOM template that is then eluted from the filter plate. So like I mentioned earlier, this occurs on the filter plate. The incubation is at 37 degrees for about 45 minutes, excuse me. Once the samples are eluted off the filter plate, we then move to the index PCR amplification. And here, as it's depicted in the schematic, we are adding our dual index barcodes. And what this allows for high multiplexing, as we can see with 96 samples. And basically, you have your region of interest, and you have a unique barcode flanking the 5' end and the 3' end of your insert of interest. At the end of this, we clean up the libraries to remove any primers, any excess material, to have a final library that is can be used for sequencing. Once we've proceeded through all the steps, that's essentially how the libraries are generated. It's a very quick and fast workflow, very straightforward. Once the libraries are generated, we want to make sure we're doing proper quality checks. And so one of them would be to run the libraries on either a 4% agarose gel, a bioanalyzer, fragment analyzer, or an instrument very similar to these to see if we are seeing the expected library at the expected size. And also it's a great check to see if there's any other additional peaks that are showing up that really shouldn't be there. Here I have an example of the control DNA. You should run, when you run this on a gel, you should see one bright band that corresponds to the library. In this case, it's about 350. For the TruSeq Bovine Parentage panel, the expected library size is somewhere around 250, as you can see here with this bioanalyzer trace, with the upper and lower marker, with that sharp peak at somewhere between 250 is our library. So once we have our libraries generated, cleaned up, we've checked them for quality, we're now well on our way to putting these libraries on the sequencer. And we recommend that libraries are normalized to the same nanomolar concentration. And this is because it gives every library within a pool an equal chance to bind to the flow cell. And if we envision here the schematic where we have 96 different libraries that were generated, each one is a little bit different in concentration, we want to be able to normalize these to the same concentration. And traditionally, we recommend quantifying in triplicate with qPCR, then calculating the dilution, and then manually dilute and pool each of these libraries. As you can see, if you've ever done qPCR, it can be quite tedious, and it can actually take quite a long time to do this manual normalization. One nice thing with the TruSeq Bovine Parentage panel is that we provide reagents to perform bead-based library normalization, which I have depicted here. Basically what happens is those libraries that are post-PCR cleanup, we run them through a separate set of beads that essentially saturates and captures around the same amount of library for each, same concentration for each library in a pool, and that is simply eluted out. Once you have all those libraries that have been normalized, you simply take 5 microliters of each library to go into your pool. This takes about 1.5 hours. As you can see, it's a lot of time saved using bead-based normalization. And it is as simple as pipetting 5 microliters of each of your libraries. So a little bit more on the bead-based library normalization. A couple of things to keep in mind is that we want to make sure that the beads are completely resuspended, and it's very critical. These beads tend to be a little bit more sticky, and so always make sure that we have properly vortexed them until all of the beads are in solution, and that we follow the normalization procedures within the user guide for best results. Another key point to keep in mind is after the libraries are normalized with bead-based normalization, they're single-stranded and ready to load onto your instrument. And so if you decide to run these on a bioanalyzer or fragment analyzer and don't see anything, don't worry, they're single-stranded, and the bioanalyzer will pick up double-stranded DNA. And because of the flexibility and the ease of bead-based normalization, you have the capability of pooling 96 samples or libraries per sequencing run. So now that your libraries are normalized, we're now proceeding on to making our pooled AFRICOM libraries to go onto the MySeq cartridge. So to pool the libraries, we simply take 5 microliters of each of the normalized libraries to create what is called a PAL. And this PAL is the pooled AFRICOM library. Once you have the pool of your 96 libraries in one Eppendorf tube, you then dilute that with HT1 to create the diluted AFRICOM library. At this point, we do a brief incubation at 95 degrees for 2 minutes and a gradual cool down. Because I had mentioned earlier these libraries are single-stranded, this heat will prevent any type of, or dissociate any secondary structure, that may have formed. And at this point, you load it onto your MySeq and you're ready to go. So once you have your reagents, your library all loaded up into the MiSeq cartridge, we then proceed on to sequencing on the MiSeq. Like I mentioned earlier, you have the capability of pooling up to 96 samples in one MiSeq run. One piece of information that you will need to be able to successfully sequence these libraries on the MiSeq is that we will need a manifest. And this manifest will be, is available from our support site for download. An additional key piece of information or software that you will need is Illumina Experiment Manager version 1.12, excuse me, that just came out recently to be able to set up your sample sheet using the TruSeq Bovine application within Illumina Experiment Manager and generate your sample sheet. Here I have an example of what the sample sheet should look like. I know it's quite small, but you guys have the slides. Just it's a nice way to double check that your sample sheet is set up correctly. And one thing to keep in mind, this is a 1 by 121 run with 2 index reads. So once your sequencing run is completed, we then use the MiSeq reporter that's on board the MiSeq instrument using the TruSeq AFRICOM workflow to then take those sequence files in FASTQ files and generate them into genome BCF files. So MiSeq reporter will align sequencing reads to the reference genome and perform the variant calling. And so the required files for this particular portion of the protocol is the bovine genome, which we don't need to worry because it already comes pre-installed on the MiSeq, and the TruSeq Bovine Parentage manifest file, which we've already downloaded when we set up the sample sheet. So the key output file that we need from MiSeq reporter and using the TruSeq AFRICOM workflow is our genome BCF files. And these files can be found within the alignment folder, and I have the file path here. And it contains all reference and alternate bases. So once we have our genome BCF files, we then move on to our new tool, which is our sequence genotyper. And so the sequence genotyper will convert variant calls from genome BCF files to make genotyping calls. A couple of things to keep in mind when we're installing sequence genotyper, we want to make sure that we have the appropriate computing requirements and software requirements. One thing also to keep in mind that the sequence genotyper is not supported on virtual machines, so this may be something that you may want to consider when thinking about where to download and install sequence genotyper. Sequence genotyper, it allows you the capability of creating different projects, importing samples and target files, and to generate reports. The required TruSeq Bovine Parentage panel target file, already comes pre-installed within sequence genotyper, so there's no need to upload a different target file. So once you open sequence genotyper, this is the dashboard. This is your home page for sequence genotyper, and here you will see a variety of different tabs. You'll be able to view how many projects you have, view your target files, samples, and reports. To create a new project, you simply click on Create Project. You will see the following pop up. You name your project, give it a brief description, and then you're prompted to the next window, which is creating your report. Here, you simply drag and drop your genome BCF files from the alignment folder that was generated with MiSeq reporter, and you drag and drop them right here, and where you see the little cloud with the arrow. Once you have all of your genome BCF files uploaded, and you will see that the upload's quite quick, and you will see some confirmation that the genome BCF files have successfully uploaded. Once you have all your samples uploaded into sequence genotyper, you simply click Generate Report, and you have the capability of giving it a name and either aligning to the top or the plus strand. Once you start the report, this is the type of output that you will see on your screen. Here at the very top is the report, and here it gives you the report ID, the report strand, how many targets, how many samples were analyzed, and how many failed calls. You will also see what your call rate is for this particular set of samples. If we go down to the bottom, you'll see on the left-hand side, you will see the list of the target SNPs, their location on a chromosome, coordinate, reference allele, alternative allele, and what the call rate is. And then you start seeing your samples. If you have, here I only have 3, but if you do have more, if you envision you have 96 samples, you will then use the buttons to the left and right of the the white box that represents the different pages that there. You just have to scroll through to look for your particular sample. If you wanted to, one nice feature of the sequence genotyper is that you have the capability of downloading these reports in a variety of different formats. And one thing also that I forgot to mention is that if you have any no calls, those will automatically get filtered to the top of the list, so you'll be able to see exactly which calls did not produce, which samples produced no calls. So like I mentioned earlier, you have the capability of downloading Excel-friendly reports, either very complex reports or simple reports, and that is simply by clicking on the Open Report button at the top. You will then be prompted to select which type of report you want to download. The matrix report is a very detailed report that includes the genotype quality value, depth, and variant frequency. Or if you simply just want to see allele 1 and allele 2 for each of the samples and targets, you can then download the simple report. These are both downloadable as .csv files that are can be opened with Excel. So just to summarize the overall workflow of TruSeq Bovine Parentage Panel, it encompasses 263 SNPs we've talked about today. 200 SNPs are ISAG and the rest are disease-associated or inheritance SNPs. The library prep follows their TruSeq Custom Amplicon version 1.5 workflow. It requires 50 nanograms of genomic DNA. We have the capability of sequencing up to 96 samples on a MiSeq run using V3 chemistry. It uses MiSeq Reporter to generate the genome BCF files that can then be imported into our sequence genotype analysis tool to convert them into genotyping calls. So now I want to move into what are some key steps to have a successful library prep with our TruSeq Bovine Parentage Panel. And so here I have a brief overview of the different steps that we've talked about today within the library prep workflow. And I want to highlight some key steps that we want to particularly pay attention to have a successful prep. One of them, we'll talk about the importance of proper quantification of the genomic DNA. Also, key steps within the workflow to keep in mind for successful prep, and also best practices with library normalization and bead handling in general. So like I mentioned, one big key component of a successful prep is proper quantification of the input DNA. We recommend a fluorometric-based method to quantify the DNA, like I mentioned earlier, either qubit or picogreen. And we want to make sure that we assess the DNA quality. You can measure 260, 280 using a nanodrop, and the ratio should be between 1.8 and 2. If you would like, you can also run your DNA on a gel to make sure to see if it's smeary in any way. If you do any of these assessments and have any questions, please feel free to reach out to tech support. We're happy to review any gels or any quality DNA issues. We're here to help, so we want to make sure that you have a successful starting material to go into the prep. And lastly, that we are using the recommended amount, which is 50 nanograms. So once we have the 50 nanograms of genomic DNA, what are some key points within the workflow to keep in mind for a successful prep? One of them, like I had mentioned earlier, is pre-washing of the filter plate. This is another key component for a successful prep. Pre-washing the filter increases the ability of the DNA to hybridize. It checks the integrity of the filter membrane. And unbound oligos, if we don't pre-wash the filter, you run the risk of unbound oligos to remain if the filter is not pre-washed. So you don't want unbound oligos at the end of your prep. It's also, if there's any wells within the filter plate that don't seem to be filtering very well, it can sometimes be very tricky because of the wells being the filter plate being very shiny. So when you're looking over it, it can be a little bit tricky. That's okay, we understand that. If you have any questions as far as making sure the filter plate is filtering properly, or you have any concerns that you have a few wells within the filter plate that aren't filtering, please give tech support a call. We're happy to help. So some best practices when working with filter plates if you're new to using this particular type of filter plate workflow. Like I mentioned earlier, the extension ligation happens on the filter plate, and we now need to elute that extended ligated product off of the filter plate. And in order to do that, we use sodium hydroxide. So one key component to keep in mind is that the pH of the sodium hydroxide, if it's anywhere under 12.5, that can definitely impact the ability of the samples to be eluted properly off the filter. If the pH is too low, that can impact elution. Or if you use too high of a concentration, more than the recommended 50 millimolar of sodium hydroxide, that ends up, you have too much sodium hydroxide in your final product, that can then impact downstream PCR. When we're adding the sodium hydroxide to the filter plates, again, I know it can be really tricky being so shiny, we want to make sure that we're adding the sodium hydroxide to the middle of each of the filter plates. And this ensures that the sodium hydroxide saturates the filter plate and is incubated and that we are incubating at room temperature for at least 5 minutes to allow for maximum dissociation. We also want to make sure that we're pipetting the sodium hydroxide up and down sufficiently at least 5 or 6 times when we're adding to the filter plate and when we're eluting the samples off. Another key component is, there's a lot of different reagents that are being used within this protocol, a lot of different equipment. So following best practices with handling reagents and thermocyclers, just one thing to keep in mind. We want to make sure that we're following best practices with all of the different reagents. If you're not going to be processing 96 samples at once, we want to avoid multiple freeze thaws. There are a lot of enzymes in our reagents, so like any enzyme, it does not like to go through freeze thaw cycles. That can definitely impact performance. If you foresee yourself not processing 96 samples at once, perhaps you want to aliquot the reagent so that you minimize those freeze thaw cycles. Another key component is that we want to make sure that we're adding the reagents in the order that is listed in the user guide. I know from my time in the lab, if I could do a premix, I would. In this case, definitely recommend following the order that is listed in the user guide. And there are some reagents that can be a little bit viscous within the workflow, so we just want to take care and pipette viscous reagents properly. Be a little bit patient as you're trying to pipette those very viscous reagents. We are using the thermocycler. So best practice is anytime you have an incubation in a thermocycler that we have the heated lid turned on to at least 100 degrees during the incubation. And this prevents condensation of the lid. And if we have condensation, that can impact the enzyme efficiency, or you end up having too high of a salt concentration. And just, a nice visual of when we have samples on a thermocycler with a heated lid versus not, you can clearly see condensation has formed. So we want to avoid that. Heated lid, always. There are a lot of bead steps in this particular workflow. So one key thing to keep in mind when working with beads is it's all about ratios. So if you have any excess beads on the outside of the tip, that can impact how well the site selection occurs during the bead post-PCR cleanup. So one thing to keep in mind is with all of our beads, all of the beads that are used within the protocol, we want to make sure that those beads come up to room temperature before use. We really want them at room. Cold beads will definitely impact the performance and will definitely have an impact on your yield. So take the extra time, put them out on the bench to get them up to room temperature. And also use freshly prepared 80% ethanol for all of the washes. The beads look like mud. As you can see here, there's very dark brown consistency, very viscous. So we really want to make sure that the beads are vortex and completely resuspended. Invert the conical tube or tube that you have it just to make sure there isn't any beads at the bottom of the conical tube or of the Eppendorf tube. We want to make sure the whole thing is completely resuspended. Other thing is that we're following and using the recommended plates and the magnetic stand. Although it seems very trivial, a magnetic stand is a magnetic stand. We have seen time and time and again, different magnetic stands can definitely impact the yield. So recommend the one that is listed in the user guide. and pipette up and down to minimize any potential bead loss, and be very careful with that. Drying, while we're drying the beads off at the final step of most of the bead cleanups, we want to maintain the plate on the magnetic stand to prevent any potential bead loss due to electrostatic forces. Now, during library normalization, these beads are a little bit different. Like I mentioned earlier, they're even a little bit more viscous than the ampere beads, so we want to make sure that those beads are fully resuspended. And what are some best practices that we want to keep in mind when preparing going through library normalization? Reagent limiting reactions required addition of equal amounts of beads to samples, so we want to make sure that we're adding the appropriate volume of beads and making sure that we don't have any excess beads on the outside of the pipette tip. How do we do this? We make sure that the beads are resuspended exactly as directed. We want to verify the shaking speed and time, and we highly recommend that a high-speed plate shaker that can reach 1800 RPM is key. We've had customers call in asking, I don't have a plate shaker that goes up to 1800 RPM. It only goes to 1200 RPM. Can I just increase the time? Anecdotally, we have seen that definitely impacts the normalization. So we want to make sure that we're using the recommended speed and time for the plate shaker. And another key thing, they both look very similar in the ampere beads and the library normalization beads. They're both that very dark brown color, so we want to make sure that we're using the right beads at the right step. So we're using the ampere beads for post-PCR cleanup, and the normalization beads are used for the library normalization. If you swap them around, that definitely will have an impact on your library. Other things to keep in mind, other best practices to keep in mind when going through the protocol is that using the correct centrifuge speed. For that filtration step where we spin the filter plate, we want to make sure that we're spinning at 2400, we're spinning at 2400 G, not at 2400 RPM. This can lead to non-specific products, and the products, your final libraries can appear smeary. Sorry about that. Other things to keep in mind is that we are using the correct sodium hydroxide concentration. It's pretty important for proper elution off the filter plate. And that we're using both sets of index primers. You need to use both an orange and a white capped index primer tube, otherwise you won't have a complete library. So we want to make sure that we're using two primers. Recommended equipment is also a key thing here. Using the Hybex heat block for hybridization, we have seen customers try to use a thermocycler even set to the slowest ramp setting. This will definitely can impact the ability of the probes to hybridize and the efficiency of that. We want to make sure that the heat block cools down gradually. We want to ensure that at least 80 minutes, it takes at least 80 minutes to cool down from the 95 degrees down to 40. And because that can be a lengthy amount of time for samples to be incubating, we want to make sure that the plate is well sealed. We want to avoid any type of evaporation. So we want to make sure that the recommended aluminum foil seals are used and ensure that those are tight. You cannot have seals that are too tight. So always make sure that we have a tight seal on those aluminum foil. Any loose seals can, as you can envision, can lead to evaporation, and that can definitely impact library prep. So just to summarize some of the best practices that we talked about today, we want to make sure that we're following the protocol as written. We want to make sure that we're following the prevent PCR product contamination that are listed in the user guide. We want to make sure that we're taking care when adding viscous reagents. We want to make sure that we complete all the recommended wash steps that are within the workflow. We're following best practices with our beads, and that we have a heated lid during any thermocycler incubation. And lastly, I want to leave you with a few resources that are related to the TruSeq Bovine Parentage panel, the support page. The support page has a lot of great information, including the user guide, protocol list, protocol guide, where to download the manifest, how to download sequence, where to download sequence genotyper, Illumina experiment manager, and I'll also have some great FAQs there that if you have any questions, you may be able to find them there. And also if you want a little bit more visual as far as the TruSeq custom AFRICOM workflow that the TruSeq Bovine panel follows, we do have some great online training that covers the TruSeq AFRICOM workflow if you wanted a little bit more detail. And again, any questions throughout the workflow, whether it's sequencing, whether it's library prep or analysis, feel free to reach out to tech support, shoot us an email, give us a call. At the end of the day, we want you guys to succeed. So give us a call if you have any questions.